Why do we age? The answer changes everything. You already know you're going to age. You've seen it happen to others, and maybe you've noticed the first signs yourself. The reason seems obvious. The body simply wears out. But that's not what biology shows. Right now, your body is repairing damage, replacing worn-out cells, and fighting decay every second of every day. So the real question isn't why we age. It's why those repairs eventually stop keeping up. The answer has completely changed how scientists understand aging, and it suggests we've been asking the wrong question all along. Start with a number, around 50. That is how many times the average human cell can divide before it permanently stops. In 1961, biologist Leonard Hayflick discovered that human cells did not divide forever. They slowed down, stopped, and never started again, even under perfect conditions. It was as if they were counting. Decades later, scientists found the reason. At the end of every chromosome are protective caps called telomeres. Each time a cell divides, those caps become slightly shorter. Eventually, they are too short to protect the DNA, triggering a simple instruction: stop dividing. This is the telomere clock. It reveals that aging is not just random wear and tear. Older cells do not stop because they are exhausted. They stop because they have received a precise biological signal. And once you realize our cells follow instructions, a new question emerges. Can those instructions ever be changed? But the story doesn't end with telomeres. They explain why cells stop dividing, but not why the body itself grows old. Because aging isn't just about cells shutting down. Sometimes it's about cells behaving badly. Scientists call these senescent cells, often nicknamed zombie cells. They've stopped dividing, but instead of disappearing, they remain in our tissues, releasing inflammatory chemicals that damage the healthy cells around them. Young bodies clear these cells efficiently. Older bodies don't. Over time, they accumulate, contributing to problems throughout the body, from aching joints to weakened immunity and cognitive decline. Then came a remarkable discovery. In 2011, researchers at the Mayo Clinic genetically engineered mice so their senescent cells could be removed. The results were striking. The mice stayed healthier for longer, developed age-related diseases later, and maintained the physical abilities of much younger animals. That changed the conversation. Senescent cells weren't just a byproduct of aging. Removing them actually slowed the aging process itself, suggesting that aging might be something we can intervene in, not just endure. That discovery changed how scientists think about aging and led to one of the field's most influential ideas, the information theory of aging, proposed by Harvard researcher David Sinclair. The theory starts with a simple distinction. Your DNA is the hardware, the genetic code that builds your body. Surprisingly, that code stays largely intact throughout your life. What changes is the software. Surrounding your DNA is the epigenome, a layer of chemical instructions that tells each cell which genes to turn on or off. It's why a brain cell and a liver cell, despite having identical DNA, perform completely different jobs. Over time, those instructions become distorted. Stress, environmental damage, and even the process of repairing DNA gradually introduce errors. Cells begin to lose their identity, and the body's signals become noisier and less precise. Seen this way, aging isn't primarily a failure of the hardware. It's a corruption of the software. And that's a profound shift, because unlike broken hardware, corrupted information may one day be restored. In 2023, David Sinclair's lab published a remarkable result. By delivering just 3 genes into the eyes of old mice, researchers restored vision loss to age-related degeneration. The DNA had not changed, and no new cells were created. Instead, the cell's epigenetic instructions were reset, allowing them to function like younger cells again. The real significance was not about eyesight. It was what the experiment suggested about aging itself. If our cells still contain the information needed to function as they once did, then aging may not be a process of irreversible loss. It may be a process of forgotten instructions. And if those instructions can be recovered, then some aspects of aging might one day be reversible. Now for the reality. None of this means aging has been solved. Mice aren't humans, and treatments that show promise in the lab may take decades or may never become safe therapies. The biology is exciting, but the timeline remains uncertain. What has changed is the question scientists are asking. For generations, aging was seen as an unavoidable decline to slow, not a process to understand. Today, evidence suggests aging is driven by biological mechanisms, and mechanisms can potentially be influenced. Telomeres can be extended, senescent cells can be removed, and in carefully controlled experiments, parts of the epigenome can even be reset. Whether these approaches will meaningfully extend healthy human life is still unknown. Researchers don't all agree, and that's exactly what you'd expect at the frontier of science. The uncertainty isn't a weakness. It's the natural consequence of exploring one of biology's biggest unanswered questions. We began with a simple belief: the body wears out. But the body isn't just another machine. It's a self-repairing system, shaped by billions of years of evolution. The real question isn't why we age. It's whether aging is an unchangeable law or a biological process. Because laws can't be rewritten, biology can. That question is no longer philosophical. Scientists are testing it right now. And for the first time in history, the answer isn't already known.